Professor Laskey, thank you so much for coming to be interviewed on eCancer TV. Really lovely to see you again. Now, you and I know about your work, but people watching this program don't. You've been interested in the area of improving detection of cancer. It's not a very sexy area. It doesn't get nearly as much money as drug treatments and translation. But quite frankly, the main issue in life is getting patients early in the cancer proliferation, because that's when you get the best cure rates without a shadow of doubt. The surgeons still cure far more patients, assuming they're early, than do the medical oncologists. That's the situation. So you've been looking at proliferation markers for some time now and looking at them with a view to getting a handle on early diagnostics. Would you like to tell us about them?
Yes, the premise is exactly the point you've made, namely that by detecting cancer early, the cure rates are far more successful. So we've been trying to see if we could identify malignant and specifically pre-malignant cells in body fluids by the rationale that's used for the pap smear test for cancer of the cervix of the uterus, but to see if we could extend that to other types of tissue, recovering cells from the surface of the tissue or from body fluids. And the first example we've looked at is to try to improve the pap smear test itself by adding an antigen against a marker that is present in proliferating cells, and a marker that therefore should not be present in the cells that are sampled in the smear test, because it should be broken down before the cells ever reach the surface, as cell division is going on in deeper layers, not at the surface. So the markers we've used for this, we tried several, but the ones that performed best in our hands were called mini-chromosome maintenance proteins. These are proteins that coordinate DNA replication and largely regulate it, but they do so from the position of being very abundant proteins in the cell nucleus. So they make very good markers. And they turn out to be expressed in the deeper tissue, in the deeper layers of the tissue where proliferation is going on, but completely absent from the surface in normal tissue. However, with pre-malignant lesions and malignant lesions, they persist to the surface and they're present in the cells that are sampled. So therefore, rather than staining all the cells, normal cells and abnormal cells, with similarly same color, but subtly different shapes that require years of training to spot the difference, we can stain the abnormal cells a different color, making them much, much easier to detect and potentially allowing automation, as well as potentially increasing the sensitivity of the test by making it easier to detect the abnormal cells and the slides easier to read. That's the rationale.
I remember you able to pick up these MCMs in urine and in
yes.
So what other body fluids?
We've looked at several other body fluids as well. So urine is one, where there have been several trials, both by immunocytology of staining the slides and biochemical slides by former colleagues, biochemical methods by former colleagues of mine. And that's one that is still certainly in progress and looking promising, but needs more research done on it. And two which we've persisted with, which are also looking promising, of a sputum test for lung cancer. There it's difficult to substantially outperform consultant-led cytopathology, but of course a color stain can be read by a machine rather than by a highly trained consultant pathologist. So that I think has some promise. The other promising application that we're really excited about is a screen for colorectal cancer by recovering cells from the surface of feces specimens and staining those. And my colleague Nick Coleman, who's been absolutely essential for this work and has been the pathologist leading our cancer detection studies, Nick's lab have now developed a method to improve the detection of colonocytes about 30-fold. So the small clinical trial that we published in Lancet several years ago, we're now ready to do larger trials because we can get back 30 times more cells than that very promising trial. We didn't take it forward further at the time because we needed very high cell yields to make it a robust enough test. So that we're certainly persevering with. So far as the cervix is concerned, I think there's a lot of potential there too. Firstly, Becton Dickinson have licensed cancer research technologies patents and are making really quite exciting progress and have produced one product called ProXC on the basis of that, which is an MCM-based antibody test. But in addition, we've done trials with the Kidwai Institute in Bangalore, who've been seeing how it can be used with manual tests. And that was published, I think, 2 years ago now, but looking really very promising that it did pick up. There were 9 disputed cases in that trial, which the MCMs called positive and the pap smear test called negative. All of those were followed up, and all 9 turned out to be false negatives of the pap smear test, and none of them was a false positive by the MCM test. The MCM test had called it correctly.
I mean, that's immensely exciting, and particularly for countries like India, who have masses of patients with cervix cancer, and I'm sure that that you'll be welcomed with open arms, and to do bigger field trials. I mean, for the developing countries, of course, cost is going to be an issue. But presumably, if you're going to be able to automate this, then you're going to cut costs.
Well, the thing
in a big
that also
way.
encouraged us enormously from that Indian trial was the fact that it took 1/5 the time to read the slide, and therefore there's a saving in costs because of the
Doctor
time. Just even read manually rather than automatically. That time saving means that it becomes really quite affordable.